Okay, I think we can start. So welcome back, everybody. I hope your day with your working groups went well, but was productive. So I have to say that so we all live in a pandemic and it's terrible, but there are few positive aspects, and one of these is having experts talking from the other side of the world. And in this case, I'm so happy to announce Emiliano Santornecchi, who is a professor at Harvard in Boston. And he is an expert in non-invasive brain stimulation techniques, which are used in combination with other neuroimaging techniques like EEG, MEG, and fMRI. And today he will talk about cognitive fingerprintings and the human brain. So thank you, Emiliano.
Thank you, Georgia, for the introduction. First of all, thank you for having me. This is really, I agree that this is a great, let's say, a silver lining of the pandemic. We can actually be more connected in a sense. It forced us to find other ways. So it's good. So should I share my screen? Can I? No, you need to enable screen sharing.
Davide?
Done it.
Great.
You're now able to.
Yes. Okay. Can you see the screen? I guess I will go just for this so that you can see the screen, even if I go. Okay, this should be fine.
Perfect.
Can you see it well? Okay. So again, thank you for having me. This is really great opportunity to talk about some of the work we've been doing on cognitive fingerprinting using different techniques and modalities, as you will see in the next few slides. For me, this talk is not about showing basically what we have done. It's more of a provocative talk about what we should do in the future and what are the opportunities, but also at the same time the challenges. So what we haven't done and why, basically, and how to fix those problems. I decided to make the talk this way so that it's not just a boring summary of what I've been doing about this topic. It's more about what are the new things and how a new generation of scientists should actually approach this problem. So it will fit, I think, a different kind of audience, people that are expert, that are already working on this, people that want to approach the field. So obviously it will not fit perfectly for everybody, but I hope it will be still a good point, like a starting point for a discussion, basically. So 30 seconds about who I am, just a bit of a narcissistic moment. I'm, as Georgia was saying, I'm an assistant professor of neurology in Boston. I have my appointment here, but at the same time, I'm still very linked to my original university of Siena in Tuscany, where I still co-direct a laboratory there. That's a laboratory where I graduated, and the lab was funded by Simone Rossi. So this is just to say that, yes, I'm on the other side of the Atlantic, but at the same time, I'm still very present in Italy. And that's why I'm really happy to give this talk today, because my activities in Italy, I would say they're really probably 50% of what I actually do in terms of research. And the other 50 happens in Boston. I have also other appointments in Boston. I'm an affiliate associate professor at the Department of Physics at Northeastern University. I have many interests, as you can see here, so my affiliation reflects kind of that. And that's why you will see in the presentation, I like to always keep a very, very open mind about any problem, and I always try to prioritize multidisciplinary work, especially when it comes to fingerprinting, understanding the human brain, and so on. In terms of what we do in my lab, we do a bit of everything. So we do functional, structural, any type of neuroimaging. We also do a combination of different techniques. So we leverage, in this case, you can see TMS-EG approaches. We do computational research, computational modeling. Again, I try to combine all these things, as you can see in this slide, to get the best we can from different fields and different expertise. I started my career, my journey, studying human cognition and cognitive enhancement. At the beginning, it was just neuroimaging. I was just trying to understand the correlates of being smarter or having a better working memory or executive function. Then I discovered non-invasive brain stimulation, which adds another tool, which is to me a fundamental tool to go from correlation to causality in a sense, and test cognitive models with this technique and understand if it's really true that these three regions explain variability in X or Y. Now with brain stimulation, we can actually test that. And then I've always been fascinated by complex network theory and complexity in general. So I always try to take a step back and not just look at the data based on the first results you get or the very high level kind of mapping that we can do with neuroimaging, but I try to go more deeper. At the same time, in the last few years, I've tried to leverage all this knowledge for translational neuroscience, meaning that I also run clinical trials in Alzheimer's disease, brain tumors, and other pathologies, trying to leverage all this understanding of the human brain and cognition and cognitive deficits to develop either new biomarker or new therapeutic applications for patients. Now, I decided to start the presentation with a few slides, actually from a presentation I gave a few years ago. It was a TEDx talk where I was looking at, when I was trying to explain the complexity of the human brain and the brain connectome. And I always start with this slide. So who are we? That's the point of this conversation, this talk. We want to understand who we are, why are we different, and how can we capture these differences in cognition and how the brain is structured and so on. A bit of history, just to create a bit of context of what is the challenge that we are facing. This is the original fingerprinting idea, to look at DNA and to find specific aspects of each one of us that are unique and can explain a lot of the traits and features that we show at the behavioral level, cognitive level, and so on. To give a quick recap, everything started in 1953. As you know, first evidence of a structure of the DNA. Only 50 years later, there was a public announcement that maybe we were really close to mapping the human genome, which gives an idea of the scale of how long it took to actually achieve this. Is that enough to look at DNA, to look at other behavioral features? In my opinion, it's not. And I think that's why a few years ago, almost 20 years ago, the Human Connectome Project started, which was a parallel to the Human Genome Project. But now the idea is that we finally realized that there is way more complexity in the human brain than you can find in any other biological feature that you can measure. And so it was really time to actually look at the brain in a new way. And so this let's say initiative, the Human Connectome Project, is just a flagship that launched this new wave of interest in the brain, which was crucial, but now it's paralleled by many other even better, I would say, initiatives that are out there. The thing about the Human Connectome is that it finally gave access to tool and techniques to really look at the brain in details. So you can look at brain structure, brain function, the organization of networks. And this is amazing when you enter the field and usually get really attracted by these crazy, beautiful, brainy pictures. But obviously the situation is more complicated than what we might think. I always like to put things in context also when it comes to a connectome, using this example. So in 1998, Brenner, which then received the Nobel Prize for these discoveries, finally mapped the connectome of C. elegans. Now, even though it can seem complicated in this graph, but the C. elegans, we all know what it is. I mean, it's a soil worm. It's probably one of the simplest thing you can map a connectome of. And it's only composed by 302 neurons and 7,000 synapses. So it's quite simple, actually, in the context of what we try to map today. And the project took 12 years to do this. Now, if you put things in context, when you look about the human brain and understand the complexity of the brain, and then everything that comes from it, including cognition, you're talking at a different scale, from 300 neurons to more than 100 billion neurons, 130 trillion synapses, which is the equivalent of the number of stars in 1,500 Milky Way galaxies. And if you want to put it even more in context, I did the math a few years ago, and it still holds up. To capture the complexity of the DNA, if you do it complete sequences and everything, you can get about 5 gigabytes of raw data. So in a current iPhone that you can buy with, let's say, 100 gigabytes of memory, you can store around 26 DNAs. So what seemed to be a challenge that was impossible to solve a few decades ago, we can now actually do and store for many individuals in one phone. If you want to do the same for one single human brain, if you do the math, you actually need 1 trillion gigabytes or 62 billion iPhones. So the gain that we are facing is completely different from what has been done before. And so this is both kind of inspiring, which could give us many opportunities to look at something so complicated that we can really think of extracting amazing new information about who we are and why we operate the way we do, why we get pathologies and so on. But at the same time, it kind of put us back to the ground and make us think, okay, this is really going to be a challenging task. And I think this is an important message for whoever wants to approach connectomics, studying the brain and so on, because we get so fascinated again by these quick, immediate results that you can get nowadays with any type of analysis and any freely available data sets, that we keep forgetting about the complexity of what we're measuring. We don't want to publish oversimplistic interpretation of what we find just because it's easier today to get nice, flashy pictures of the brain and brain activity compared to what it was just 10 years ago or 15 years ago. So going now in cognition, I will just show you what my journey has been very quickly. Basically, we can look at many things when it comes to cognition and doing fingerprinting, or at least understanding individual differences. One aspect, and this is the only slide I have about structural features and structural correlates of cognition. This is a work that we published, I think, 1 year ago, 2 years ago. We look at how different features of the brain, meaning thickness of a specific area, surface area, so how big an area is, the convolution and complexity of this region, how that translates into specific cognitive functions. And you can still find this kind of information there, even though you're just looking at the morphology of the brain. In this case, we found that for certain type of skills, more related to abstract reasoning, when you do hypothesis testing, it's better to actually have more like a bigger region, because you need to navigate a parameter space that is bigger and you need to do multiple hypothesis testing. So that's what you need. While when you go into something that is more similar to, let's say, crystallized intelligence or knowledge-based cognition, you actually need basically more pruning process to refine the region that support that function. And so the region is actually smaller, but it's actually, in a sense, more efficient, and you have less cortical columns, but those columns are more efficient in doing the specific knowledge-based task. This is just an example of what we can do to find difference between individuals in terms of specific cognitive functions. But to me, structural connectome and cognitive differences has never been a crucial aspect of my work. I think we can go beyond that, and that's where the vast majority of the field has gone. And mostly it's about studying spontaneous brain activity. Fascinating fact for those of you who don't work on this kind of data. Why did people got interested into spontaneous brain activities? Because basically, when you think about this concept of how much energy, metabolic demand the brain requires to operate at rest, it's kind of fascinating to discover that it requires basically 20% of the overall metabolic demand that we have in our body. So it's a very energy consuming organ. And even to do nothing, to rest on a beach, like you can see in the picture, it requires quite a lot of energy. Now, you would imagine that if you move from the picture on the left to the picture on the right, so you start, I don't know, driving an F1 car or be an astronaut in space or do a surgery, your brain should completely change and reorganize. And basically, it shouldn't look anything like what it was before. And there should be a demand in energy that would be, I don't know, 5, 10 times, 100 times more than not doing anything. But actually, the surprising fact is that when you look at metabolism, there is only an increase of 5% of this energy to do whatever is on the left to compare it to whatever is on the right. But actually, it's not 5% of the total 100%. It's 5% more of this 20%. So basically, it's something about 1-2% more. And just with that 1-2% more, we are able to do this compared to this. So there is something beautiful about this spontaneous activity of the brain, which seems to be already enough to determine what we are able to do in a sense, and we are prepared to do based on how our brain is structured and how it oscillates at rest. That has started the field of connectomics, basically, and now we look at many networks, resting state networks. You can see here, probably many of you are familiar with all these networks. We know that this region tends to oscillate and synchronize at rest spontaneously. We are trying to understand, we are starting to understand, in my opinion, why they do that and what are the implications of these networks in pathology and in explaining cognitive differences. But in my opinion, I mean, I always try to leverage this to understand, again, cognitive differences. And these are some of the work that we published over the years. I will show you details on a couple of them, just as an excuse to show how I approach this problem of fingerprinting and cognitive neuroscience and cognitive differences. These are some of the work. I use EEG, fMRI, other techniques. So we put a lot of effort on this, and the results sometimes are quite interesting. In this work, for instance, we look at the role of strong and weak connections in the brain. So we wanted to understand why everybody always look at strong connections in the brain. That's the typical approach. When you do a connectome, functional connectome, you usually filter the number of connection and only look at the strong one, which you can see here in red. But obviously, you always cut these weak ones that you tend to think are kind of noise almost in the brain. And actually, when you look at other fields like biology, what you discover is that in many other complex networks, weak links, weak connection are actually the most important one because they stabilize the network compared to the strong ones that are definitely important for information flow, but the weak ones have an equally important role. So in this paper, for instance, we look at different type of connection. We went from the very strong one to the very weak one. And we found that, for instance, IQ is more correlated with weak connections that you can see here, and a bit with the strongest one, compared to what we thought before, because before the analysis were only done at this level by looking at strong connection. And so this was another way of getting inspired by another field and trying to understand individual differences. And when we look at what these connections are, to our surprise, the weak connections in the brain are actually connections that are not well organized. They kind of connect everything in the brain, from the right to the right hemisphere, different lobes. It's almost like a weak network that keep everything together with low metabolic demand, which to me was quite surprising, again, because we usually look at these connections in the brain to define cognition and correlates of cognition. Another example, we look at, for instance, why do intelligent people live longer? It's kind of a stretch as a concept, but this is a known fact. And probably there are many socioeconomical aspects that determine this and many other things. But we also want to try to parameterize this thing looking at connectomics. And so we actually start looking at this connectome and start lesioning the connectome in different ways. So we want to see if the brain of someone with a higher cognitive profile can afford getting more lesions, for instance, than the brain of someone with a lower cognitive profile, which is a new innovative way to look at the connectome. Instead of just mapping it, we now play with it and we lesion it. So we did two type of lesioning. In one case, you just randomly lesion the network one by one, and every time you measure how resilient, how efficient the network still is. In the other one, you go directly for the stronger nodes in the brain, the most important region from a functional perspective, and then you go down until you reach the one that really don't seem to matter much. What we found, again, to our surprise, is that there is a relationship between your IQ that you can see here on the x-axis and the resilience of your brain. So the higher the IQ, the more the brain can afford a higher number of lesions before losing efficiency, which gives us an explanation of why, again, people with higher intelligence, together with many other factors, as I said, might actually be able to afford more lesions, which lesions could be strokes or any other type of lesion, go into surgery and not develop delirium, for instance, after surgery. So this is another spin that I like to give to the study of cognitive differences, just to give an idea of how we can actually look at things from a different perspective. And we were also able to actually identify the regions in the brain that actually provide more resilience in people with higher IQ and high resilience in people with lower IQ. So you can also get quite specific about deciding which region to look at. Last example before jumping into the main paper about fingerprinting. This is work done by Arianna Menardis. She's a student in Padova with you, a PhD student. She just spent 1 year with me in Boston, and we did this study looking at the genetics and connectomics and cognition. So we want to understand if there is a relationship between these three things. If you find something, in this case, executive function, that correlates with some aspect of the connectome, like with a specific network, can we find a genetic component? Can we see if that's due to, again, DNA, it's a heritable trait or not? And so in this case, we analyzed 450 twins. All of them has functional imaging and executive function assessment. We mapped their connectomes, and then we basically look at how resilient is their brain were when we actually do the lesioning problem process that I mentioned before. And so we look at how different networks related to different executive function. In this case, we have a common executive function factor, shifting and updating, how the brain of these subjects actually respond to different lesioning process. And we find something quite interesting. We found that 3 networks that support executive function actually show some mild irritability. And you can see it here, monozygotic twins versus dizygotic twins. You can see that the difference between the 2 siblings is way lower in the way their brain responds to lesioning, the region that correlates with the executive function response to lesioning in the monozygotic twins compared to the dizygotic twins. So this is another kind of insight on how we can try to bring everything together and study cognitive difference, not just by looking at behavior, but actually adding connectomics up to adding basically genetics and do everything as a multi-level analysis. But this is the real paper that everybody wants to look at when we talk about brain functional fingerprinting and the relevance for cognition. This is a paper published in Nature Neuroscience, I think in 2015. It was still based on the human connectome data. So they look at hundreds of subjects. They find different networks, as you can see here. They define the network for each subject. The main story in that paper, which was very fascinating about individual differences, in that the authors were able to identify a set of regions that are either highly unique. So this connection between different lobes, you can think about this is the frontal lobe and the posterior part of the brain. Some connections are very unique. So these are the connections that every individual tends to have different from the rest of the sample, while there are a set of connections in the brain that are very consistent. And these are usually related to sensory systems. So you don't look at this region probably to find some specific feature of each single individual. The interesting thing is that then they try to predict fluid intelligence, in this case, abstract reasoning, one might say. And they found a nice way to predict this based on the overall connectome by looking at each individual brain and characterizing each individual brain, or by looking just at a specific network, the frontoparietal control network, which seems to be the one that really predicts best when it comes to cognitive function in this sample. But the most important piece is here. So they were able to achieve, when they were given enough time points, enough data, they were able to achieve 100% accuracy. I think it was 98% accuracy in differentiating and identifying each individual in the sample based on their functional connectome. So now the functional connectome becomes literally like a fingerprint. So every subject has a unique connectome. And if you have enough data, you can actually identify one subject out of 1,000. But the most important piece for me was this result, which was not very stressed. But in this sample, they had both task-based data. So patients were doing a task inside the scanner, and it can be a memory task. One was a motor task. So it's a typical way to look at cognition with fMRI, for instance. And then they look at resting state data. And as you can see, resting state data, even though we like to think that it's the new thing, it's amazing, we can find a lot of information there, really didn't discriminate much differently than the evoked data. So when you combine the two, you actually get close to your 100% accuracy in identifying each subject. So this is a way to say that resting state maybe is not enough. And we need to look at other ways to actually capture individual differences in the brain. Obviously, the best is to combine the two. But if you want to go after one unique approach to do this, in my opinion, the way to look at that is by perturbing the brain connectome, which is what I'm going to explain now quickly, which is where I'm investing the vast majority of my effort now when it comes to cognitive neuroscience and understanding differences. So what I mean by that, I mean that basically we know from physics that if you want to capture information about a complex system like the brain or any other complex system, you get way more information if you actually perturb the brain and you look at what happened afterwards than if you just look at the brain, the complex network at rest, like when it acts spontaneously. So it's like throwing a rock in a lake and look at the ripples. So what we conceptualized a few years ago is that when you actually stimulate one brain region, in this case with magnetic stimulation, you can actually look at how activity propagates in different parts of the brain. In this case, we use EEG. And if you have a region that is very connected to this stimulation area, you will have a nice, strong response, which happens right after the perturbation that you induce with brain stimulation. If you have a region that is very connected to the same stimulation area, but in another part of the brain, you should get a very similar response because they're part of the same network probably. Instead, if you have a region that is not really part of the same network, you might have a different response. It can be weaker, it can be delayed. So it's not really sampling the same thing. You can also have, as an extreme, a region that is not part of this network and is not even talking with the network. And so you don't see any difference before and after you send a TMS pulse, a magnetic pulse. So to me, this is a kind of a game changer. That's what I call a perturbation-based biomarker to study cognition. And we also use it a lot in my clinical trials to understand difference between patients, but also longitudinal changes after an intervention. Can be a drug, can be brain stimulation or anything else. For those of you who don't know what brain stimulation is, I will keep it very simple. We have different types of brain stimulation that we can do. The big difference is what we call transcranial magnetic stimulation, which is what we use for this kind of perturbation-based biomarker studies, and also transcranial electrical stimulation, which is what I prefer when it comes to actual therapies. TMS has been out there for almost, I would say, 35 years now. So it's very validated. We have FDA approved for treatment of various pathologies in the United States. It basically sends a strong magnetic field that translates into an electrical field in the brain. It can actually make neurons fire. So it makes you able to induce the activation in a region. And now you can causally prove that when you activate that region, something happened at the behavioral level. So the way we do TMS-EEG to study cognition, we basically zap one region, in this case, let's say the motor cortex. But the advantage is that when you keep an EEG cap on, so you have electrodes on the rest of the scalp, you can actually measure this TMS evoked potential called TEPs, each one from each electrode. So you get a nice mapping depending on how many electrodes you have, or what happened in the brain when you stimulate one region and how the signal propagates through the brain and so on. So to me, it's a super valuable tool. And one example, if you want to take a look at one paper, is this paper we published earlier this year in PNAS, when we look at individualized perturbation of the human connectome to basically determine and identify potential biomarkers of network dynamics and also their relevance for cognition to see if we can capture individual differences with this. So the way we did it very quickly is we stimulated two brain networks. I would say two of the most dominant one, the default mode network in red and the dorsal attention network in green. And the dorsal attention network, you might know a lot about this because you have the world leading expert on this system, Maurizio Corbetta, there in Padova. So we stimulated these two regions, and the idea was that we can actually look at how the signal spread when you stimulate one network, let's say the green one, the dorsal attention here, or the default mode network here. And we can do it in each subject, in each participant. So I will just show you one example participant to see how the signal spread. The beauty of this is that these are milliseconds. So now you see the TMS pulse happening here. There is some activity propagating in the brain, but it's very close to where we stimulate. Again, here, maybe moves to this other node, come back here. It doesn't spread much. It's a different story. And again, these are just 50 milliseconds that you just look at. So compared to fMRI, we can look at high temporal resolution data. The different story is when you stimulate the other network, the default mode, which is more integrated. It has nodes in the prefrontal lobe and the frontal lobe. So you can see the TMS pulse now, and now things propagate in the brain, which is very different. It's a more dominant network, I would say, at rest. So when you zap this network, you get more propagation. It's a different trajectory and so on. So why am I showing you this? Is because we then basically look at the individual differences in how each brain responds to this perturbation. When you stimulate network A, that was my original question, is it important how much you engage network A But I think it's even more important to look at how much you engage network B. So you want to see some specificity. If your brain is well segregated and your network are well defined, when you stimulate one network, you shouldn't see a lot of bleeding of signal into the other network. So we call this network engagement specificity. And we found out that people with a lot of network engagement specificity basically tend to elicit more activity in the target network, in this case, the green one compared to the red one, and vice versa. When you stimulate the red one, you get more activity in that one compared to the other one that you are not stimulating. But you also have subjects that don't really modulate, don't really have a difference. And this can be due to the fact that their network are too connected one to each other. The brain is not segregated enough, and other scenarios like that. But the most important piece is that when we try to correlate all this with cognition, including the resting state EEG that we have on this sample, we found out that when you look at resting state EEG, which you can see in black, the correlation with two types of abstract reasoning, logical and relational, IQ, and motor task, for resting state EEG, so look at spontaneous brain activity like we do with fMRI or resting state EEG, didn't correlate much with cognition. But actually, when we correlate this effect, so the network specificity after perturbation, we see the correlation going up, especially for a couple of scores, including IQ. Another correlation moved from almost 0 to 0.5, 0.6. So the idea here is that when you zap the brain and you look at the response to the perturbation, you actually get way more information about each individual brain that you would get just by measuring the brain at rest. And this, in a sense, applies also to fMRI studies that are very similar results there that were published before our study with TMS EEG. So for me, this is a way to do a quick assessment with TMS and EEG and really capture individual differences in cognition. But the idea is that you actually need to introduce a perturbation to do that. So it's something that definitely we should consider in the future of cognitive neuroscience. To go even further, we are doing actual fingerprinting of this TMS EEG response in these subjects. And the way we do it is basically looking at different sites that we stimulate, like 5 sites in the brain. And we look at this is the clear concept of why we need to do fingerprinting. So we look usually at the grand TPs, the average of all the TPs that you get from all the subjects in your group. And in this case, we also have test retest reliability. So we did the same assessment with TMS EEG twice, at 1 month distant. So when you get these as a result, you might think that you are getting some TPs, so you see some potential. But what you don't see is that when you look at each individual subject, like subject 1 and subject 2, what they show is that across visits, like 1 month after the first visit, you do visit 2, the potential that what subject evoked are kind of similar across session. It's kind of surprising, like 1 month apart, different time of the day, like 1 month apart, everything can happen in that month, but you actually get a nice similar response. The same for subject 2, more or less. But when you compare subject 1 and subject 2, they don't look anything similar. So you're stimulating the same region with the same machine, with the same setup, but each brain responds differently to the perturbation, to the point that when you actually use our TP measurement for the 2 study visits, you get 100% accuracy in identifying each individual subject in our study based on how they respond to the perturbation. Each brain does a different thing. But when you look at actual standard measure, like global mill field power or spectral power of the resting state EEG, the accuracy is only chance, 50% accuracy. So it's another proof that actually zapping the brain provides way more valuable information about each individual subject. And when you look at which region of the brain is more important to capture these differences across brain, we discovered that, not surprisingly, left dorsolateral prefrontal cortex is kind of the dominant one that produces 80% of accuracy by itself. And then the other regions tends to produce a bit less, but cumulative, we get 100%. This to me was a very fascinating result that goes in the direction of doing the same fingerprinting that they publish in Nature Neuroscience with fMRI, but doing it with TMS EEG. Last thing I want to stress is that to me, even though we do all these complex multimodal studies, TMS EEG, imaging, and so on, to me, the models and the prediction are still too static, simplistic, and semi-unimodal. So what I mean by that is that I think we need to always keep in mind when we do the studies that any finding we see is not really definitive, and it shouldn't be, because now we have the tools to look deeper in the brain. So this is an example of a study, a grant, European grant that we just got called NeuroTwin. We got it a few months ago to create basically a virtual brain And this virtual brain would include everything from micro level to mesoscale and macro scale. What I mean by that is that we want to reconcile everything that happened at the level that I just showed with TMS-EEG or fMRI, which is micro scale, is interaction between regions in the brain or networks. But we want to go deeper and look at mesoscale. So what happened in each individual node of this network, how can we capture the activity of different cortical columns, how we can we capture the activity of the different neurons and interneurons in the cortical column that generate the oscillations that we just look at in the EEG. And then we want to go down to micro basically. When we look at the activity of each individual cell, and with computational model, we can recreate the activity of these cells and then create oscillations and recreate EEG and so on. So this is something I think everybody that approach cognitive neuroscience and want to look at individual difference, use imaging or electrophysiology should always keep in mind. I mean, whatever we find, there is always something deeper that can actually flip the interpretation of your results if you actually go to this level. I know it's not easy, but I think anybody that approaches this field should always keep in mind that this is going to be the future at some point. And for me, it's already the future. And so you always need to think in that direction. Even then, you just stick to macro scale. It's perfectly fine, but you need to always reconcile your finding with previous literature about something before that. At the same time, everything I show you now, apart from the TMS-EEG data, is based on, and I would say the 90% of the cognitive neuroscience literature is based on static assessment of brain functional connectome. We call it functional, but then we average everything that happened in the fMRI scanner into one single connectome, one snapshot of brain activity. But we know that activity in the brain change every second. And with TMS-EEG, I just show you that it changes every millisecond. So I think it's time to really stop looking at an average of activity over a 20 minutes period or 7 or 10 minutes period, because we know that the brain fluctuates across networks, across brain states, and every brain state is supported by different networks. So I would say it's more misleading to look at an aggregate like this matrix over time than to not say anything. Because when you look at the average map, it can be the summary of 5 different states that happen at 5 different times, and all in a different way in all the individuals that you're actually measuring. So you don't know really why this metric looks like this. It can be the same metric for 2 subjects, but it comes from completely different dynamics. So another thing to consider is not just the spatial resolution, but also the temporal resolution, and just start looking at dynamic functional connectivity, like Gustavo Deco did in this paper. This is another example of this kind of work. I mean, this is what I'm trying to do to contribute to this field. I'm working with mathematicians to actually build predictive model of spontaneous brain activity. We use differential equation to build a model that look at how each region oscillates and why regions in networks tends to be either in a competitive state or in a cooperative state. And based on that, we were able to build, for instance, a model that predict resting state functional data for a few seconds, I would say a couple of minutes after the moment when you get the baseline data, basically. And this model is actually cool for me because we go back to the lesioning approach I showed before, and it allows me to now play with the brain as a toy model and predict what happens if I actually modify the brain and I introduce a lesion or I change a link in the brain. And now I can run a simulation like what happens if I change the default bone network, and this is the original matrix. This is how the matrix looks like now, and these are the connections that needs to change to adapt to this perturbation that I introduced. If I do it in a more cognitive network like the dorsal attention, this is the original matrix. Now this is what happened, and the connection that needs to change, but are different from this one. So for me, it's a new way to look at the brain as a tool, basically, to run also simulations. Last point, and then I'm done. I think it's crucial, and this was at least my experience, to open your research to other fields. For me, the journey about understanding intelligence, individual differences in intelligence was an exciting one at the beginning. I tried to use every technique, EEG, fMRI, DTI, structural connectome. But at some point, I lost the value of the journey because I was always looking at the same type of techniques like neuroscience tools. And I decided that I need to expand my horizon, and I actually need to get inspiration from other fields. And that's what I did, for instance, for the lesioning and resilience approach. all those knowledge about how to lesion networks and the resilience of networks come from physics and mathematics. When I look at weak connection, that kind of inspiration came from biology, and mostly evolutionary biology, and the fact that you see the same importance of weak connection in other complex systems and in the macaque brain and so on. So what I did in terms of intelligence, I actually built a consortium, which is called the Neuroscience of Intelligence and Cognitive Enhancement Consortium, the NICE consortium, where I basically bring together experts from any other field that I couldn't get access to. I mean, I work with machine learning, but evolutionary biology was really out of my field. We look at plant biology, the intelligence of plants, social intelligence. We look at everything that up to philosophy that can inspire us and understand what is intelligence, what is higher order cognition in humans, and not just from a nice kind of neuroscientific framework that always leads to these nice, bright, shiny figures that give you the impression you're understanding something, but I always like to remind myself that probably I haven't. And so in this consortium, we have many faculty members from all around the world, advisors, and we try to do it as a team, which is my let's say, final advice for this presentation, to always try to go beyond. So to recap what I just showed you, I know it was a lot, but also nothing, because it's difficult to summarize cognitive fingerprinting and cognitive difference in nowadays neuroscience in 20, 30 minutes. But I think the point is that, yes, we can capture individual differences in cognition via structural and functional imaging. I would say also with electrophysiology. Next generation of scientists, researchers should always look at novel way to capture these differences. We have been doing this one-to-one mapping, like correlate the brain connectome with one cognitive score for 20 years now. I think it's really time to think beyond that. And in my experience, multimodal approaches like TMSEG or computational models are key to increase temporal resolution, spatial resolution, but also treat brain connectivity as a state, not a trait. So to look at what changes in the brain every second, every millisecond, that's where even more differences will be found. Because in the original paper here, which got 100% accuracy, they were just looking at an aggregate, the average activity over 2 or 3 resting state scans. They weren't even looking at how the brain changed during those scans. And was the same for us, for TMSEG. I mean, we had to do some sort of an average, but imagine what you can get if you actually now start looking at how each brain is different in terms of second to second or millisecond changes. This is basically the same message. Never stop questioning your findings. That's what I just said. I'm fascinated about always being wrong and always thinking, okay, I see this thing at the network level, at the cortical level, but why is that? I know that it's publishable and it's definitely a good incremental step in the literature, but I really invite people to think about things at different scales, even though then you publish the paper as it is, because it's the most effective way to convey your findings. And last slide I show, get inspiration from other fields. To me is key to understand cognitive differences. I know it seems kind of a stretch, but when you start digging in, you actually understand like why it's valuable. And the last message is still from my last old TED talk. Always remember that if the brain was so simple we could understand it, it would be so simple we couldn't, which is a nice paradox that always brings me back to earth in a sense, and always remind me that we always have this impression we're understanding. Now we have all the tools, all the imaging data, the data sets, but the quest, it's really an unprecedented one. And so we always need to be cautious about what we publish, what we say, the interpretation of the findings we have. It's really like a core mission, I think, for the modern neuroscientist to always take a step back and don't get driven by the appeal of publishing findings just because how easy sometimes it is to find cool findings. So that's the end of the talk. Again, I don't know if it was enough, but I really like to think about this as a first step for a conversation. And if anybody has questions or wants to collaborate or talk about this stuff, I love to talk about this for hours or days. So this is my email. Feel free to send me an email.
Thank you, Jan, for your terrific talk. It's very fascinating. Are there any questions from the audience? So I have a general, obviously unanswered question because it's just a provocative one. So you started your presentation showing us how complex is the brain. There are millions, billions of neurons, trillions of synapses. However, there is this new trend in neuroscience showing or suggesting that we are more similar than we thought. So there is a low dimensionality in brain and behavior. What is your position, your feeling? How can we combine these two paradoxical aspects? So on one hand, we have a high degrees of freedom, but on the other hand, a low number of components can describe our behavior or our brain activity.
Yeah, no, it's a very good point. And that's the type of conversation I like to have. It's more of a philosophical, conceptual approach. Like, should we waste this much energy into understanding every single bits of the brain, or can we stop at the level where we need it? Like, to understand cognition, maybe you don't need to map all those trillions of connections. You need 10 millions and you're fine. So I think efficiency is always the key. If I don't think we should overspend energy, waste energy on understanding something that is this complex and invest this much effort. So we need to be smart. That's the goal here. At the conceptual level, I think it's still risky to go for something that looks like a simplification of the problem because it gets appealing. It's like the same thing I was saying about brain mapping. As soon as we discovered that we can actually map this network and you can map it in 5 different ways, you start seeing 5 papers that show the same thing with 5 different tools to show the connections, because we think, oh, we got it. This is a slight difference from the previous finding, but it's crucial. Like, our brain it's attracted by this kind of simplification because it makes us, in a sense, happy and fulfilled with what we do. So conceptually, I would say we always need to keep an eye on how the brain is complex, because you might find something when you actually look at all those trillions of connections or neurons that flip everything you thought about. It's unlikely, but it might be there because we don't have a clue of what happened at that level. At the same time, we need to be practical, and that's how we push science forward. So that's the message at the end. I mean, always keep in mind the micro scale, the meso scale, but if you have a nice, cool macro scale level, network level finding that fits and can inspire other people to move forward, publish it. That's what we did with the TMSG paper. I don't think we understand anything about the brain does those things, but it seems to be valuable for cognition. So I hope someone else will get inspired and do that.
Thank you, Emiliano. There is a question from the messages. I read this. Noemi asks, was the correlation between networks and connectivity and IQ positive or negative?
So let me think. This is about IQ positive and negative. Is this a weak and strong connection paper?
No,
I don't know which
it was
one.
The paper about the IQ and the individual variability about the I don't know how to explain. One of the paper of the middle in the middle of the discussion.
So one of my papers or other papers? Just to
I think your paper about the individual measure of the individual measure that correlates with IQ.
The resilience measure, when we remove nodes from the brain?
No, the resilience, the fact that stimulating one network will affect the other network.
Okay, so the TMSEG study,
yeah,
the one with brain stimulation. Okay. So what's the correlation between networks and connectivity and IQ, positive or negative? I don't know if I understand what you mean by networks interconnectivity in that study. So maybe I get it. So you mean that difference between the network we stimulate and the other one? Is that what you mean?
No, I don't remember the name, the acronym of the measure. NAR.
The network engagement specificity.
Yeah.
NES. Okay. Yeah. That
How
one.
correlates with? the IQ.
Was okay, I got your point. That's the crucial message of the paper. Sorry, I didn't even mention it. So this is a great question. It was positive. So the more specific is your response, the higher your IQ, which fits with the idea, the correlational evidence that we have from previously in the literature that says that a more segregated brain, when every network does what it's supposed to do and only talks with the other one when it's needed, even though they always talks, but let's say in a more structured way, that correlates with the higher cognitive profile. So in our case, when you zap on network and you only want to engage with one, if you don't engage anything else, you have an higher cognitive profile. So your brain, we think it's a reflection of the fact that your brain is segregated. And when you stimulate one network, the signal only propagates within that network and doesn't go everywhere. Okay. So the correlation is positive.
Now it's clear. Thank you.
Sorry.
There is another question, a more practical question, but very important also to me. So with a multidisciplinary approach to neuroscience, how can we be sure we are relying on the right source? Do you personally have a routine when you decide that it's time to get inspiration from different scientific fields and whom to trust?
Do you have an answer for that? I think it's a question for everybody. It's always a risky proposition to go out there and try. I'm extremely curious, so I come always from a place where I've already read more about what my results look like. Like I always get attracted to that kind of thing. And so I get a sense of who are the experts in the field, who I think fits with my approach to problems, the kind of publication that people make. And then I kind of approach these people and we start talking. Usually I don't approach these people when I have a result. Meaning I ask them to validate it or not. It's at the very beginning of the process. So it's a conversation you start and you say, hey, you published this thing about evolutionary biology and how the brain actually evolved to have more important weak connection than strong connection. We don't look at that in the brain. Is it valuable? What do you think? I did this with the author of that paper, Semerly, who is a mathematician from, I think, Bulgaria. And he has this theory about weak connections in complex network. He never thought about the brain. And when I present my idea, I was like, oh, you can measure weak and strong connection in the brain. That's awesome. Let's do it. So it's a bet. You go and you hope that you find the right people. I don't know if I'm answering your question. It seems you have a very, very specific question about this. So I don't know if you had bad experiences doing that or, but yeah, it's a challenge. You always need to find the right people, I know. I don't know if I answered the question, otherwise you can follow up with another one.
Very good.
Yeah, maybe it's easier if I follow up with my voice.
Awesome.
Yeah, the thing is that I find it really hard to communicate with different experts, let's call it experts in other fields, because maybe sometimes I mean one thing with a term and then they mean another thing. And then, yeah, there was a practical question on how to speak with those people. And if you have some suggestions.
I mean, it's about studying their field. So first it comes with some effort from your side. You need to read those papers. I remember the first time I read the paper from Laszlo Barabasi. Now he's a collaborator. I'm an associate professor in his lab. He's the godfather of complex networks. Like he defined a complex network in connectomics in the 90s. Is a genius. I read this paper, I remember on the train from Siena to my place in Italy for probably 2 months straight, and I didn't understand anything. And then it clicked, and I was like, okay, now I understand what he's doing. So then I start applying his methods into my paper. And now finally, when I went to talk with him in Boston, because he's here as well, we spoke kind of the same language. You need to know enough to have a conversation, but then you are talking with an expert, so they are supposed to guide you on many of the aspects. And I think that's the key. You need to study, and the fact that it's challenging, it's part of, I think, being a science and promote progress in science. It's going to be a lot of extra effort. You could do it without. You could just stick to your modality and your expertise and do it. I think it's a great contribution to the field. If you are the one making a connection with, I don't know, crazy one, an astronomer that look at the structure of galaxies, I think that's worth a career. If that leads to something important, but it takes you 2 years to understand why that's valuable, that's why we are here for. What we are here for. So I know it's painful. I'm just saying it's a way to look at things and how to promote the field in general, it's not mandatory. So I know it's supposed to be tough, Andrea, so there's no clear answer. Just keep pushing.
Yeah, it's worth it, I think. Thank you.
So one curiosity, did your measure of intelligence had the component of creativity too in it? Because what I was thinking is, when you zap a particular part of the brain and when you see certain networks getting activated and you have associated that with intelligence, right? That specific network. But when we observe creativity, it's not really necessarily true. And but when you see an activity, that's what I think. It's my impression that when certain areas, for example, if you are thinking about a flower and you get an idea and maybe an idea, mathematical idea, and certain other part of the brain, and what I feel is in this case, the association will be inverse, in where if you activate a particular part of the brain, the other part of the brain should be also getting activated in terms of creativity. So I'm just curious whether your investigation just was limited to intelligence or it also had the component of creativity.
So that's a great question. We didn't consider creativity in the study, but we do a lot of research on that. So we have actually tried to find the commonalities and differences between the creativity network, insight network, and intelligence network. Let's assume there is a network. We just look at the meta-analysis of activation and so on. So we are trying to understand what are the overlaps, and when you engage one network, what system can you actually also get effects on the other one. In this paper, we didn't look at creativity because we were stimulating networks, in this case, dorsal attention and the Faulkner network, which we and other teams have previously published as being correlated with the type of measurement that we were doing, like fluid intelligence, abstract reasoning, and so on, and IQ. So for me, I have a couple of publications, maybe 3, on the role of the dorsal attention network in fluid intelligence and abstract reasoning as the most dominant network. The Faulkner network has been associated with everything, in a sense. So we had to pick something high level to test the framework, like they did in the Nature Neuroscience paper when they look at fluid intelligence and very high level stuff. It would be, I think you're absolutely right. Depending on where you stimulate, you might see a strong correlation for some function and not another one. And that's when imaging can guide where to stimulate. I think that's why it's a nice circular thing. We have a look at creativity.
Right, that will be interesting actually if we try to probe the area. Thank you so much for your wonderful.
That's the advantage of this technique. No, go ahead. Yeah.
We have time for just one.
Yeah, there is a question from Francesco. So you mentioned a way to analyze cooperation and competition between different networks. Can you briefly explain what cooperation and competition mean when talking about brain networks activity? Does it mean correlation, anti-correlation, something like that?
It doesn't. So that's another beautiful example of where talking with other people that have different expertise really helped. So the competition versus cooperation model comes from evolutionary game theory, which means that it's game theory as we know it. When you really look at players in a game and you want to know who cooperates, who doesn't, and why we see the dynamics we see and how decisions are made. Evolutionary game theory actually look at how these things change through time. And so we were able to build a model with differential equations that they model that. And basically it models the metabolic demand of each region in the network that you select. In that case, we map the entire brain. And you look at how much these different regions in the brain fight to get more metabolic energy. And you map it using the Bolt signal. And so you see that some regions has to cooperate, so they don't fight each other. You can model this mathematically, while sometimes they enter in a state that when they compete, and you can determine a payoff. So for each second of recording, you can determine what is the payoff, what is the advantage of being cooperative or competitive in the next time point. And if you do that, you can predict that certain region will be more cooperative in the future, less cooperative or more competitive. And you can model that. And when you do that, you actually get your nice fMRI time series. So you can summarize and simplify all these oscillations in the brain as a continuous fight over energy in the brain. And sometimes regions are kind of smart and at times to cooperate to get the energy, sometimes they fight. And if you just use that simplified approach to determine the complex behavior, you actually recreate nicely the time series. And I wouldn't have never thought that if I didn't have experts in Siena, actually in Italy, working on evolutionary game theory. And to go back to Andrea's question, we spent 1 year just talking about this stuff on a whiteboard and we couldn't produce any result. It was super frustrating. And then we got the breakthrough and we looked at the prediction and we could predict 2 minutes. And I was like, wow, it's painful, but it's worth it. So it's not just negative correlation or positive correlations.
Okay, thank you.
Thank you very much, Emiliano. That's been a very interesting discussion. Time for the next speakers. Next speaker, Sebastian. Sebastian, are you here?
Thank you.
Thank you for being so patient to wait. Sebastian gained his PhD in neuroscience
I'm here.
from McGill in 2020, this year. He's from Germany originally, but is now in Plotly since October 2020. And he works at Plotly in Montreal, Canada. So he's too from the other side of the Atlantic Ocean than Europe. Welcome, Sebastian. Thank you very much for joining us.
Yeah, thanks for having me.
You should be allowed to share your screen.
Yes, I think I am. Okay, can you see my
Very
slides?
good. We can.
Great. All right, yeah, thanks for the nice intro. My name is Sebastian. As was just said, I have a neuroscience background myself, and one of the things that I've done during my own PhD was hang out at many BrainHack events. So I definitely appreciate being invited back. It's always great to do some neurohacking. And in my own work, one of the things that I often found challenging was to display the complex results that we often come up with at the end of, months or years of work in a single figure, right? So if essentially, in a way, I guess, one of the things that we're producing are scientific visualizations. And it's hard to bring across all of the information in just a few panels. And so one thing that I found very helpful and that now is supported by some journals even is to use interactive visualizations, just for exploring my own data, but also for sharing with other people and giving the reader essentially the ability to explore on their own some of the results. And so from that work, essentially, came my interest in working with Plotly, and I've joined that company in October of this year. And what I'm doing there is I'm working with a team that is being led by Emmanuel Gouillard, who's one of the core developers of the Scikit Image library. And this project is being funded by the Chan Zuckerberg Initiative for Essential Open Source Software. And among its goals are things like this, where I'm talking to people in the neuroscience or life sciences community to both tell them about what Plotly has to offer as tools for your visualization needs, and also to get feedback. And some of the questions that you might have are also interesting to me and to the team. So the two things I want to talk to you about today are Plotly Express and Dash by Plotly. And so they serve slightly different use cases. And the thing I want you to take away from this talk essentially is an understanding of when is it a good idea to use them? What are they meant for? How can they help you essentially? All right. So I'll start with Plotly Express. I'm not sure if some of you have already used these tools, but I'll try to keep the big picture things short and then just jump into the demo. So the idea behind Plotly Express is that it allows you to very rapidly iterate on a single figure. So the general scenario here is you have a large data set that's tabular data. And for example, you might be working with some of the more recent large neuroimaging data sets, and you just look at the phenotypic information. It's a huge table. You want to get a sense of that data. Then Plotly Express is a tool that lets you very quickly explore this in an interactive way. And you can iterate to a point where you've generated some insight, and maybe this is something that you actually want to include in a publication. And Plotly Express supports that. You can export these figures. They will stay interactive. You can share them. And as I said, some journals even let you just put them in the paper as a replacement for static figures. It's an open source library. It's under the MIT license, so you can go ahead and use it in any way you wish. And yeah, I'll just jump right in and show you how that works. So for my demo, I'll be using the Jupyter environment. I'm assuming that you're all familiar with this or you've heard about it. If not, Jupyter is a really cool interface to work interactively with code and to do some quick prototyping and have your figures generated in line. And so Plotly is going through some effort to make it easy to develop things in Plotly, in Jupyter environment. However, if you are somebody who likes more like an integrated development environment like PyCharm, VS Code, these tools also work there. Right. So the coding demo I'll be showing here is kind of domain general, but I'll show you some app examples later that have more of a life science focus. Just a quick question: can you see the font size, or do you want this a little bit bigger? Well, I'll expect you scream at me if you don't see it. Okay, so the first thing I'll do is I'll get some of the data that comes with like some of the example data. And what we're looking at here is data from the Gapminder Foundation. And that's just a data set that shows us demographic information and economical information for many countries and across several years. So if we want to get a sense of what we have here, we can see there are many years here, and the most recent year is the year 2007. So if you're familiar with the syntax, the data that I've just loaded here is just a pandas data frame. And pandas is a really great tool for tabular data in Python. And so what I want to do is limit myself, at least for the moment, to data from the year 2007. Okay. So what can I do? Well, I can get started and say, okay, well, there's some variables in here that I might be interested in. For example, let's say life expectancy, right? So I use the histogram graph type from Plotly Express, and I say, okay, well, take this table and then show me the, oops, that didn't work. Show me the distribution of life expectancy just across this table. And let me just move this little thing here. Yeah. Okay, and you see it's a histogram. Seen these things before. Nothing too surprising here. There's a little bit more interactivity. You get these little tooltips with the bin sizes and the counts, and you can actually do things like zoom and zoom out. But that's not what we're here for, right? We want to do something that we cannot do with the static image. So this is where the idea of rapid iteration comes in and is really, really helpful. So one of the things that we cannot see, but we might be interested in is, well, what are the actual discrete values that go into these bins, right? And so we can say, okay, well, give me a marginal plot, and in a rug plot, just show me these values. And here they are. So we can see there's a big range, and well, here's one country that has a really low life expectancy. And you might be wondering, well, which country is this, right? So what you're seeing here, these little tooltips are called hover info, and we can use we can ask Plotly to style them with other variables that we have access to. So in our case, we can say as the hover name, which is just the title of this little tooltip, use the variable country. And you're probably already kind of getting a sense of how the syntax works, because we're just using variables from this table here, from this pandas data frame, and we're mapping them to keywords in the Plotly Express function call. And then Plotly figures out how to show them to you. So now that we've done that, we can see, okay, well, this is Swaziland, and here are all these other countries. And now we already know where the individual values come from. And that's something that's not that easy to do if you just need to filter your table. But we can keep going. We can say, okay, countries are located in different continents. Maybe you would be curious to know if there's a difference in life expectancy across continents. We could have that idea. And so if we do the color attribute here and we map the continent variable to it, so just show you again, there's the continent variable. You can see that now this image is split by continent. So both the marginal plot has been split, and also the tooltips for the histogram have been split. And we can do things like turn some of them off if we're interested in only a subset, and we can turn them on again. We can still do our zoom and all that. But now we're already getting kind of an interesting perspective on our data set and just some small function calls. And so one of the things that we might be looking at here is, well, I mean, we have all this information. There's not a whole lot you can show in a histogram by itself, right? There are other graph types that would be more suitable for some of the data. But we can just say, well, even though we can't think of anything to do with these other variables, we can just say, well, please, Plotly, give me this just as part of these little hover informations here. So what we can do there is say, okay, we'll take all of the columns in our table. Let's, I think, not the right keyword, yeah. And so now we're mapping all of the columns in our table to the hover data keyword. And what this will do is just display them here, right? So we're not doing any plotting with them. We're just dumping them in there so we can look at them and maybe get a sense of what's going on. And if we look at this and we have a lot of time, or we have prepared this demo in advance, then we might get the sense that, well, maybe there's a link between the GDP of a country, or the economic output, and life expectancy of its citizens. And we might have also arrived at this thought in some other way. And so what's really cool is that, as I said, there are many different graph types, but they all use a very similar syntax. So we can just say, okay, well, we want to do this now as a scatter plot. And in the scatter plot, we want to map the life expectancy on the vertical axis. And then we want to explain it. I just need to make sure I get the variable names right here. We want to explain it by the variable called GDP per capita, GDP per cap. And then we don't need our marginal plot anymore, because now we're looking at individual data points. And there we go. Okay, now we have a scatter plot. And we still have our hover info. We can still do all of the other things we've just seen. One of the things we can see here, and again, that is something we probably already know, is that unfortunately, wealth is not uniformly distributed at all in the world. So one of the things that might be more appropriate here is to do, a logarithmic scale for the horizontal axis. So we can use the log x keyword here. And then with the scatter plot, we now have the ability to actually show a little bit, or encode a little bit more of the information of our data frame, right? So we could say, well, all of these countries, there's a different number of people who live there, right? Their populations are vastly different. So we can use this variable. Here it is, pop, and we can just map it to the size of our little circles here. And if we do that, we can say, okay, well, now they have different sizes. It's not very impressive yet because the sizes are really small. So we can make them bigger. And actually, since the aspect ratio of this plot is a bit underwhelming, let's make the plot itself a little bit bigger. Okay, cool. So this is already a pretty nice talk. If by any chance you have seen one of the more famous TED Talks by the Gapminder Foundation, you might have already seen this figure. So quite a lot of information in here. You can see that there are differences by continent, there are differences in population size. There's a general relationship between wealth and life expectancy. And you know, this is an interesting figure. You might just think, okay, well, I wanted to put this in my paper, and then you can export this figure. However, there's one aspect of this data frame that we're not making use of yet, and that's the historical information, right? So obviously we have information across many different years. And if we just use that information, that doesn't make for a great figure because now each country shows up many times. But one thing you can do with Plotly Express figures is you can animate them. And so what we do here is, again, the same logic. We're taking variables from the data frame and we're mapping them to these function arguments. And in this case, we're saying, well, as the animation frame, so basically the step size, we want to use the year. So our animation should increase by years. And then the thing that we want to animate, or I guess the thing that the objects are supposed to be animated, are countries. And then the last thing we have to do is to say, well, we want our plot in the vertical dimension to be predefined for 20 to 100 years, because obviously these values will change over time, and we want this to be constant. And if we do this, then we get a scatter plot, and we now have this little bar here on the bottom. We can see we have data from 1952 to 2007. And currently we're in the year 1952. We still have our scatter plot. But if we click this little play button, you can see that now we have an animation. And that animation is a really nice way to show like a time series in this case. And we can see overall countries tend to increase both in wealth and life expectancy. And that is also a figure that has been shown in this TED Talk, which is the reason why it's included in this demo. But the point of this is just to show you how quickly you can get there by doing relatively few function calls and using a relatively simple syntax. And so the last thing I want to show you from the Plotly Express side of things is something that has been more recently added to the library as part of this project that I'm also working with. And that is image annotation. And as I said in the beginning, this project is led by Emmanuel Gouillard, who is from the scikit-image library. And so we're just going to get something from scikit-image here, from, oops, import data and IO. And so as our data, we are going to use Chelsea the cat because everybody likes cats. And then to show you Chelsea the cat, I'm going to use the imshow function, which if you're using Matplotlib, you've probably used this before. And so we're feeding an image array. Here's the cat. We can see it's a cat. It's a static image, but contains all of the information. Now, if we just swap out Matplotlib for Plotly Express here, and this is something that's been recently added, now you have the same information, but you get a little bit more interactivity, right? You have the pixel position, you have the color information, you can do the zooming and the zooming out, and that might already be what you need, right? That maybe you just want people to be able to zoom into your figures and see more detail, but that's not everything you can do here. Because one of the things that's happening under the hood is that Plotly Express creates a figure object. And so you can inspect this figure object or just look inside essentially. And when you do that, you see that it's essentially a dictionary and it has different keywords. So it has a keyword for data, which includes the data that you're displaying up there. It has a keyword for layout. Excuse me, that includes information that on how the image itself is structured. And if you use a plot like this that is animated, so what we've done before, then you can see that it also, yeah, you cannot see that because it's a very large thing, but you can use this really cool feature in Jupyter, which is the JSON mode. So essentially now you, rather than just printing it, you get the interactive way of exploring this. And what I wanted to show you here is you have the data keyword, the layout keyword, and for animations, you also have the frames keyword. And this is a really cool way, like just looking and inspecting the figure object is a good way of understanding how these things work. And why would you want to do that? Well, you can manipulate these keywords, right? And so I have prepared a little bit of code, so I don't have to type everything here. But whoops, one of the things you can do is you can update the layout keyword. And what we're doing here is we change the drag mode to a drag mode called draw rect. And you might already know what that means, because if we run this now, we get the same figure. But now if I do this, I'm actually drawing a shape there. And you can draw many shapes. And one of the reasons I might be interested in doing this, I can also move them around. One of the reasons I might be interested in doing this is if I'm annotating this image, I might want to label these regions. I might want to use them in as a training set for a machine learning algorithm. So that's something you could do in an image annotation context directly in Plotly Express. And there are other drag modes. One of the things that you might have already seen here is if I interact with this image, then this thing up here appears, and that's called the mode bar. And that generally gives you an overview of what you can do with the figure, right? You can export it, zoom, pan, do all kinds of things. And I am now interested to have this mode bar display other available options. So I will just do this. So what does this do? I will take, I'm writing a little dictionary. I say add to the mode bar other draw modes. And so we've already seen draw rectangle, and here's some other ones. If I run this, now you can see that the mode bar contains some additional elements. And let's say we want a closed free form, because maybe we want to really like capture where the eye is. And maybe that doesn't work so well, but you get the idea. We have like a free form drawing tool here. And yeah, I always do this in the demo, give the cat a little piece of glasses. Cool. So this is already, I find this really fascinating. There are many things that I can think of what I can do with this, right? A lot of the work that we often do is annotating the brain or drawing regions of interest or correcting classifications. And so you might be wondering, well, I have all these ideas of what I want to do, but how do I get the information about the annotations I've just done out of the picture? Because right now I'm just looking at them, and that's cool, but maybe I have some use for them. Maybe I want to do something with them. And so this is where Dash comes in, right? So Dash is essentially a framework to generate interactive web applications, or dashboards, how they're commonly known, with like an analytical angle. So something you want to do something there, you want to generate new information, you want to generate insights. Some interactivity is important to you in order to understand something about your data. And so to do this, typically you need a number of skills that are, I would say, less commonly taught in neuroscience, and mostly because they're not that relevant for our research. So that's JavaScript, HTML, the whole web developer stack. And so the promise of Dash is that you don't need to know these languages. You can stay in the language that you're most familiar with. So let's say in my case, that's Python. This is also available for R or for Julia recently. And you can just lay out and describe these apps completely in this language environment, in Python, for example. And Dash will take care of the rest. So the tagline is no JavaScript required. And again, it's an open source library under the MIT license. And you're welcome to use it in whichever way you see fit. So let's jump in there. For this, I've created a list of little commands, so I don't have to type this all out. And if we start with a very simple app, let me see, I might have already used this port. Let's see here. Yeah, he's complaining, but that's okay. We'll just run it again. Okay, so I've run the app first, but one of the reasons that it's nice to do these things in Jupyter, by the way, is that you can see you can do this side-by-side development, which is really easy. You have the code here, and then you have the output, so to say, over there. The reason this works, by the way, is because I'm using Jupyter Dash, which is a library that is specifically made for this purpose. It allows you to prototype and serve Jupyter, Dash in a Jupyter environment. And if you were doing this with the intention of deploying it on some server, you would be just using Dash by Plotly, right, rather than Jupyter Dash. Okay, so let's take a quick look at what we're doing here. We're getting Jupyter Dash, obviously, and we're defining the app as a Jupyter Dash app, and it gets a layout, and then at the end of the script, we just run it. So the other thing we're getting is Dash HTML components. And what that does is it exposes to us the essentially HTML elements that you would otherwise write in a website. So if you've ever looked at HTML code, or if you've ever written an HTML document, you know that there are these different keywords, so to say, that tell the browser how things are supposed to look like. And so you have access to these through the HTML core components app, HTML components app. So in the layout, we don't have to take care of the entire outer aspects of this document. We just start and we say that the first element is just a div, which is like a container. And inside the div live two things. The first one is H1, which is a first order heading, and that just says hello BrainHack. And then there's another second order heading that says, look, I don't need any JavaScript. And so when you run this, as I have done, you see over here that this is what's rendered. And I mean, there's obviously no interactivity yet, but one thing I can do is I can change things over here, and then they will update over there. So this is nice, but obviously we're interested in interactivity. So let's get that. So before I run this, let's take a quick look at what has changed. So I'm now importing also something called Dash Core Components. And like Dash HTML Components gives you access to HTML elements, Dash Core Components gives you access to generally things that you can interact with, like a text field, a drop-down menu, like little radio buttons, or all kinds of things like a slider that a user can interact with, and then you, with this information that the user enters into your app, you can do something with. And the other thing we're getting is from Dash dependencies, something called input and output. And the reason we need this is because interactivity comes from linking things together as input and output through something called a callback. So if you're familiar with Python, then you will recognize this syntax as a function decorator. If not, it doesn't matter. The only thing you need to understand here at this point is that we have a little function defined up here that's called simple callback, and all it does is it takes a value as its input and it gives it back to me. And what's happening around that function, so to say, is that we have a syntax here that says, give me the element that I have defined in my app layout. So here maybe I should talk about this first, right? So here we still have our little heading, but now the second order heading that we had before doesn't actually contain any text. It just has an ID. And so the ID is the way that we can refer to individual elements. If I had more than one H2 heading, then they should have different IDs so we can differentiate between them. And the last element I have here is just an input field, and we give it the ID, the input, but it also doesn't have any value at the moment. So if I run this, I just get the first order heading, and then an invisible or empty second order heading, and then an empty text input field. And so what this callback does is it says, take the element with the ID, the input, which is my text field, and take its property value, which is essentially just what is written in here is the value property. And then feed this to the callback. The callback gives it right back to me. And then this says, take this value that is coming back, or the return of this function, and feed it as an output to the element with the ID, the output, which is my second order heading, and assign it to its children property. And so children is just a conventional name here for whatever is inside the element, and for a second order heading, that's just the text that is rendered there. So you probably already have some sense of what's going to happen when I start typing here. Well, hello, I am, whoops, Sebastian. Okay, so as I'm typing here, with every keystroke, the value is updated. This fires, and it's fed back into the second order heading, and it gets rendered directly here. Cool, so now we know how a callback works. Maybe you're interested in, well, can we do something interesting with it? I mean, this is not very interesting yet. Well, we can go back to Chelsea the cat and essentially answer this question from before: what can I do with my annotations? So what has changed here? I mean, it looks like I'm doing a lot more things, but really I've just copied over the code from the demo before. So we have Plotly Express, we have a scikit image import, and then here we get the data for Chelsea, which is just the image array, and then we make a figure out of it, we update it, we do something to the mode bar, and then in the layout we have our first order heading as always, and then as our second element now we have something called a graph object, and so it gets an ID, the graph, and a graph expects a figure, and so as a figure we pass along the figure we just made of Chelsea the cat, and also the configuration with the mode bar. And then we have a third element that is also empty initially. It has the ID, the output, and that's just there to receive text and display it. Okay, so what does our callback do? It takes data from the graph. So every time the property relayout data changes, and so relayout data is one of 4 different interactivity events that will fire if you do something with an image. So relayout data is one of them. You can select data, but relayout data happens if you change something like drawing a shape, for example. And so if that fires, we feed its contents to the callback, and then the callback is looking for a keyword shapes in the data that has been passed. And if it finds shapes, then it just takes that content and it writes it out as a JSON. So that's a way to format a string essentially. And then we take that output and we give it to this currently empty element here to display it back to us. And so what's the idea behind this? Well, how do I type something? What's the idea behind it is that now we want to actually see what is stored when we draw these little shapes here, and then figure out how we can use that. So let's say I do the same thing as before. I draw a shape, but now through this callback, I actually get the information that I've entered as a shape given back to me. And you can see that we have the coordinates of the shape. It's a rectangle. We can see that its color is cyan. We can see what the width of it is. That's a solid line. We can make another one, and then this will show up as well. Another thing that we can do is, because we've added the mode bar, we can do a freeform, and we can see that now rather than coordinates, we get a path back. And so we have access to this data. Currently, we're not doing much with it. We're just displaying it. But as my last example, I'll show you essentially a use case for all this data. And right, so now the only thing that we're doing differently is we're now also creating a second figure, which is a histogram. And the histogram, as its input, just takes the image array of Chelsea the cat, but flattened into essentially a vector. And what we're asking here is, well, just give us a histogram of the intensity values in the image array. And then we now have 2 graph elements. So the first one for the cat and the second one for the histogram. And they both come with their own IDs, histogram and my cat graph. So we link them together in our callback as we've done before. And we also listen to the shapes keyword in the information that's coming in here, but rather than just writing it back out as we've done before, we now use this information to find the coordinates of the rectangle that we're drawing, or we're going to draw, and then use these coordinates to limit the area of the image array that we want to have the histogram drawn for, right? So you can see here we're slicing the Chelsea array, and then the histogram is drawn only on the sliced portion. Okay, so if you run this, we expect to get 2 graphs, and here they are. A bit small, so I'll just take the whole screen. And now you can see there's a histogram already drawn, and we can see its interactivity. And if I now draw a rectangle, then this updates, right? If I draw it over the nose, this updates. Okay, so you now have a general sense of how to build interactivity. You can obviously keep building additional callbacks. You can keep expanding your layout. You can keep building and make very complex apps. In the interest of time, I'm not going to show you how to do that in the demo, but we have some examples. So Plotly is hosting this website called Dash App Gallery. And the idea behind the app gallery is that it's both to show you how what is possible, essentially, right? From all different kinds of fields, from business and the life sciences and so on. But it's also there to give you a starting point for your own app. So maybe you're thinking, okay, well, what I really want to do is like a little teaching demo for a simple support vector machine. And then you can just go into this app, take its source code as your starting point, and start changing it. So it's all of these apps here that the source code is available, and you're free to use them. The apps I want to show you with more of a life science focus that we've been working on in this project are, the 3 I have here. I'm going to share my slides later on. I'll have access to all of this information. And it's a little bit slow today. So this app, if you can get it to run, is, well, I can show it to you here in the screenshot. It's essentially what we have done just now with drawing shapes and annotating an image. But the idea behind it is that you can use this to generate a training data set for a machine learning algorithm, right? And so I think, yeah, maybe the server has some issues, but the general idea is that you're linking this image so that you can annotate. I mean, you can believe me that that's usually working. And then the annotations that we've just printed out as text show up in this table here that is currently empty. And the point of this demo is to demonstrate how, by linking the table and the image, you can make interesting manipulations. Let's see if one of the other ones is actually starting up. They all seem to be a little bit slow. That's very unfortunate. Well, yeah, well, this is going a little bit. Actually, the most interesting thing I wanted to show you is this one here, because to me, this is a really nice demonstration of why you might be really interested in Dash, other than the aspect of interactivity and not having to know JavaScript. So the setup of this app here is that you have the image Plotly Express ImShow that we've used before, but and you can draw shapes on that. But the interesting idea here is that the shapes that you draw are actually input features to a classifier, and this classifier is running, and it's written in Python, and it's running on the remote server. So the thing that's currently seems to be struggling. And oh yeah, okay, you're out of time. Cool. Yeah, let me just go over this really quickly. So if you click Show Segmentation, you send this back to the server, and it will send the segmentation right back to you. And the reason this is possible and is running so fast is because this code is optimized in Python, and you're running it on the server where you have a lot of resources rather than in your browser where you don't. You can do things like this, update it, and then this will update live. Okay, I won't go over the other examples. One thing I'm quickly going to touch on is all of this is obviously open source. You might be wondering, Plotly is a company, how does it make money? There's something called Dash Enterprise. It is generally targeted at very large companies, so you're probably not going to be the target audience. But if you're interested, or if you need to scale up, or have security problems, or things that you want to, you have security needs rather, then this is kind of the thing that Plotly is offering. Important thing here is just to remember Dash itself, like the Dash open source library, is not limited in any way. And my last point would be Plotly is using a lot of energy and time to write very good documentation in my mind. So you should be taking advantage of that. And go ahead, go look through the documentations. They're very interactive. And one of the things I would recommend you take a look at is the documentation on the figure structure. There's a community forum. There's also obviously a GitHub organization. So trying to be a good open source citizen. It's always nice to hear back from the community, maybe with some feedback. And yeah, my closing point would just be, there are two aspects of the world or the tools that Plotly is offering you. One are the graphing libraries, and I've shown you the high-level API, which is Plotly Express. And there is a way to tie these elements together into an analytical app with Dash, and you can do these things while still staying in the Python ecosystem that you're familiar with and that you know how to write good code in. All right, well, thank you. That's all I have for you today. If you have any questions, you can either reach me under my Twitter handle or you can send me an email. Cool.
Oh, thank you very much for your nice presentation, Sebastian.
Yeah,
the very first part actually reminded me of Factfulness by Rosling. Is this book about our idea of the world, which is a bit outdated. And he made this point by showing data and showing graphs. So it's very important data visualization for having a point in showing phenomena to a crowd.
Yeah, I think it's both fun and really useful to make good figures.
So has anybody any question to Sebastian?
I'm also on Mattermost. If you come up with something later, you can reach out to me, and I'll see if I have a good answer.
Very good. Thank you very much.
Thank you, Sebastian.
Thank you. Thanks for having me. Have a nice rest of your day.
Okay.
Bye.